Thank you all to be here. Thank you to Federica and all the team behind that decided to create this opportunity that, strange enough, we never really exploited, despite the many years several of us know each other. So I will give you an idea of what we are doing at Elettra within my activity that is related to protein production and having created a protein production facility to support research, mainly for drug discovery. So this is just to show you where we are settled. The structural biology laboratory is very close to the synchrotron ring. It's on a walking distance of a few minutes. So that means there is an exchange between the laboratory and the beamlines. And the structural biology laboratory has been completely refurbished and restarting in 2010 with the general aim to exploit molecular and structural biology techniques to study the basic genetic mechanism of the cells, and mainly through the characterization of proteins that are involved in the mechanism. And we used mainly crystallography, but not only, to determine the structure of these proteins. With the years, we exploited other methods like SAX, and nowadays, the aim is to go through a cryo-EM, where Silvia is more experienced, but we are all interested to be involved. And of course, we're settled to work in biochemical and other biophysical approaches. In the laboratory, there are also protein production platform, because traditionally, structural biology as being very heavy consumer of high quality proteins, historically became by brute force very experts in protein production. This is a kind of a settled situation, and because of this we have focused also on this activity. And of course we have a crystallization suite. In the laboratory there are two groups, the Silvia's group, who is the head of the lab, and she will explain what exactly is her work in detail in a month, I think. And well, my part is more linked with the cancer cell signaling and drug discovery related proteins. This is because I have been contaminated in a way by the industry, because during my history I grew up, I started as a medicinal chemistry, then I translated more and more toward molecular biology and structural biology. But during this story, while I was traveling a bit around the world, I passed through an industrial a big pharma at the time in the nearby of Milano, where I've been involved very much in the drug discovery process in kinases and cancer-related kinases. This is why I've been maintaining this type of attitude and interest. Within the laboratory in 2012 and 13, thanks to fundings coming from the Italia-Slovenia Interreg collaboration, we started a protein production facility with the aim to, export our skills and competency toward support of academic, industrial research and development groups in synergy with the beamlines. And so we build up all the pipeline from the initial design of the constructs, going up to the high scale up of protein production, quality control, and structural determination. And of course, being a small group, we have been focusing in working in a very collaborative attitude, working both on fundamental research project or proprietary research project that are the one coming with the industrial partners. And we have been always having growing approach of tailor sites projects. So we're working on projects, not on basically on new methods. And we have more in the years, we have seen it also a good way to support others' activities is to do tutoring and hosting students or PhD postdoc in our laboratory to get trained and start with their own protein to in the timelines of 2 to 6 months, they normally get the protein they want, and they can continue the project farther away. So in general, so we have been touching different types of objectives, of aims, working on proteins for different tasks, having proteins from projects that are from academics, but also, as I said, from industry, and starting mainly from cancer as an initial interest that was coming from direct personal involvement. We have been then transferring to our other either method development, neurodegenerative diseases, and of course, in recent time also infection diseases. Just a few words about the system of the expression system we're using to express and purify our proteins. To express the protein, we have been focusing and developing, maintaining always 3 methods of expression. One is in E. coli, very traditional, where we have developed a number of different strains. We are working a lot of insect cells, with the insect cells, baculovirus infected insect cells, and with something that we have de novo implanted in the lab, and this has been for a long time our golden standard in the expression of more complicated protein. And then we have also some experience in mammalian cell expression. In the laboratory, we have also a robotized system to, say, develop more and high throughput scale expression system. And despite we have the system there, I must admit that the end being so small, we don't have an intense work on this type of approach, mainly because of the benefit ratio between cost and benefits in the typical type of experiments we are performing. And while for the scale-up production capabilities, we are in the range of a mid-scale-up, where we go for E. coli from in the range of 2 to 10 liters with the fermenters, or normally we use shaking flask in the range of 8 liters. We have insect cells. We can go up to some tens of billion cells using specific bottles in the shaking flask, and mammalian we also have suspension culture. I mentioned this because in this respect, thanks to the PRP project, we will have some, say, improvement and upgrades in this part of the incubator system we have available. So in the sense that thanks to the project, we will acquire some new incubators. As far as regards the proteins, when we purify protein, we evaluate different characteristics from the purity level and monodispersity that for us normally being interested in characterizes the structure and going toward crystallography is an essential requisite. So we perform size exclusion chromatography. And also we have a system that is combining size exclusion chromatography with light scattering detector, so we can also detect having an information about the total mass of the system and distinguish the oligomerization status of our protein, purified protein. We have been performing also activities assays to set up a screening assay in the future and inhibition to test activity with inhibitors. This is the case, for instance, of kinases. We have the possibility to evaluate the folding status of the protein, either by using circular dichroism, that is the system in the laboratory, or using very intensively this thermostability assay. That's a kind of a very simple and analysis that one can do on the protein, evaluating its tendency to unfold with the increase of the temperature. In the presence of a fluorophore, it's possible to follow the curve of denaturation. And this is very informative of the global fold of the protein, and also informative. So, and this tells you if they say the product you have reproducibly obtaining is always the same. You can see the effect of a single mutation that will alter the stability of the protein, and what is also interesting that you can detect the binding of with inhibitors, that normally an inhibitor is attached, is binding directly, specifically with the protein, increase the number of bindings with the protein itself, and independently will increase its stability. We have also need of using mass spectroscopy. At the moment, we have been outsourcing this activity, but we are confident in the future not to have a more, say, easy access laboratory, because with the PRP project there is the investment to obtain a LHC mass system, and so Andrea is here. I know how much he's working on this, and we are confident. And the other thing is very important for us is the binding assay. For the moment, we have been doing binding assays exploiting a SPR-like system that is in the laboratory of CNR of Alberto Cassetta with Sonia, and with Sonia we have been using this system very successfully several times, but in the future we expect to have also an upgrade in this part within our laboratory. About just a few words on the crystallization suite, we do have, say, a dedicated area for crystallizing protein with an high throughput approach. You need to have to test several thousand of different conditions and using very small amount of protein, the minimum as possible. Normally you use nanoliters, so that's why you have robotics that are capable of handling nanoliters. We had also an imaging system that should have been able to monitor all these different experiments in parallel. This really never successfully worked. So far we have been always doing manual inspection, visual inspection, manual personal inspection through a normal microscope. But okay, in the future, there is the plan to refurbish and implement new instruments, both for the nanoliter robotics and the imaging system, to be capable to support better external users in the project. And twice, okay. Now, case studies, just to give an idea. I haven't tried to give an idea of the type of protein we have been working so far in these years. I will not go too much in the details and not touching only proteins that are of interest strictly with the infection or in virals and pathology and pathogens, but just to give an idea what we have more comfortable used in this in our work. So of course, because of my background, we have been working on several kinases. You know, kinases are the golden standard of the target-based drug design. Anyway, we in the recent years have been focusing, and this is part of an internal research that based on interest in autophagy and learning how autophagy how autophagy, that is, you know, it's a very relevant mechanism of the cells that is maintaining its homeostasis during stress situation where there is the evaporation of nutrients. Autophagic event is activated, and autophagy is related indirectly to several diseases, either cancer or neurodegeneration, and also in infection diseases has some roles. So we have been always interested in working a bit on this mechanism, and I've been focusing on the OLK1, that is together with OLK2, a kinase that is the initiator of the autophagic machinery. So OLK1 and OLK2 are, say, two large proteins. They are pretty homologous, and they have in the N-terminus part, it's located the kinase domain. Then they have a very long region of 450 residues of serine proline rich area, highly phosphorylating, and then the C-terminal globular domain that is responsible of interaction with all the initiator complex of the autophagy. So we interrupt by this protein. We have been thinking initially to start with in parallel and with two approach. One, thinking of the OLK1 complex, and one more drug development like, and then focusing on the kinase domain. due to different reasons and occurrence of other projects in the meanwhile, we have been just starting on the OLK1 complex, developing and preparing the construct of the whole complex, that is the initiator complex of the machinery, while for the OLK1 kinase domain, we have been go through the whole process, and this I can just show you where we have been working more recently. So thanks, this is all the work done by Marta Semrau and Theo Battista. They have developed, Marta has developed an expression system that's working rather well in E. coli. She managed to obtain good protein. She set up an assay and translated and transferred this assay to the high throughput screening facility of the Cibio in Trento, and with setting up an autophosphorylation assay, because the protein is capable to autophosphorylate itself, we could do a high throughput screening with about 4,000 compounds, of which the majority was coming from the internal library, diverse library from the Cibio department, and a KTGS library that says a very focused library already rich of inhibitors that are well validated against kinases. From this, we obtained 22 compounds. Some of them are pretty active. And so, of course, we have done then the next step. Theo and Marta could obtain crystals and solve the crystal structure. So now we are at the level to understand. We know where these compounds are binding. Not surprisingly, are binding in the ITP binding pocket, but at least it's a starting point. And we are now waiting to get some hints on the mechanism of action on the cells in collaboration with the University of Udine. Then, beside the kinases that we have always running in the lab, due to the pandemic, we have been pulled in the proteases world. So we have been, and as Electra, we have been involved in a very big project that was named Escalate, that was led by Dompe, and I must say that was also thanks to Federica that we got engaged with them, and they decided to involve us in the structural biology part of this project. And so we have been focused on the proteases. I think I can go very quick on the story of what is. So the main protease, it's very well known, a validated target. So behind this, there was all the work done by the virtual screening laboratory. We have been working quite a lot on repurposing inhibitors. That has been as all the others. So this work was done doing a classical screening of almost 9,000 compounds that were coming from a EU screening library, the Open EU library, proprietary compound from the Fraunhofer Institute, and the first and the free for men compound that were validated by Dompe. From all this screening, we obtained a number of compounds, and of few of them we could obtain a nice crystal structure at the end that were informative for the design of other compounds. So we have been focusing, the large majority were coming from peptidyl inhibitors, though they mimic the protein and the binding, they sit very well in the active site. This is MG under 32. What is nice from this story is that, okay, we simply proved it is binding. It is an irreversible pseudo-irreversible inhibitor that covalently binds the cysteine that is in the active site. But the MG under 32 was originally known as a proteasome inhibitor. And by homology modeling and from some results, we have we noticed that could have been capable of inhibiting also cathepsin L, and because of the interest in cathepsin L being also involved in the uptake of the virus in the infection, we could prove that there is a binding effect indeed, and the MG 132 inhibits at micromolar level cathepsin L, and so this can be used to improve with the idea to create dual dual inhibitors that can either bind the protease of the virus and the cathepsin at the same time. Myristatin is another story. This is a flavonoid. All the flavonoids are indeed known to be antiviral. I mean, are very often having effective activity as antivirals. This myristatin is binding quite tightly to the protein, and we could observe that the binding mode was different from what was normally found for homolog compounds with other flavonoids. So this has been then opened some ideas on how to create a better irreversible binder to this against the more specific for MPRO. So the story then continues. We are now focusing more on other peptidomimetics or also smaller compounds that are not reporting here, that have been, this is collaboration that initiated through the ESCALATE and now is continuing with the group of Vincenzo Summa at the Federico II in Naples. And so with them, we could provide basically the structural content of their design and do a proper SAR in the development, proving that indeed their expected mechanism were supported by the structures. There are some stories like this proline that they designed to be important in improving the potency. Indeed, this was the case, and so at the end we could do this part, just giving the validation of the activity. The other story regards the papain-like protease, is the other protease of the virus. This it's very interesting because it has this very characteristic thumb, palm, finger architecture that is typical of the deubiquitinases. So by itself is a cysteine protease that has this fold, and indeed in the cells it acts also as the deubiquitinase. So it has an effect in de-IgGlating, especially deubiquitinate, and especially ISG15 is a ubiquitin-like small protein that is linked with the interferon cascade and the innate immunity. And this explains why probably this protein has an effect in the infectivity of the virus, not only on its own viral cycle. Being the protein, we have been working as we normally do, starting from the construct. In this case, we have been focusing, I've been the only one to create a construct that was a bit longer than the standard, because everyone is working on the restricted protease, the PL-PRO domain, because PL-PRO is a part of a very long non-structural protein of the virus that is a multi-domain protein and contains actually as a multi-activity. It's difficult to be studied by itself in the integer form, but so we have try to add at least another small domain that is the nucleic acid binding domain. So we have used these two proteins in parallel for the screening. And this has been done also Fraunhofer with the same characteristic of the MPRO. And interesting, at the beginning, we obtained a lot of hits that resulted to be very potent compounds, and we are enthusiastic about that. Of course, some of them were nanomolar. Stefano, who has been doing most of the work, he produced a lot of crystal, but they were always apple. So it means either they were not growing, or once they could grow, they were apple. That means there was no compound bound in the active site. So what Stefano could notice is that when he was doing this thermostability assay, so following the melting curve of the protein, in the presence of these inhibitors, he always observed a shift to the lower temperatures, like the protein was more destabilized. That is pretty unusual when you have a specific small molecule that should sit in its active site. So then we started to try to understand what was wrong. If it was our assay, there was something that was not compatible with these compounds. After a lot of time, and doing also the mutants, when Stefano did the mutants, he realized that this effect was not visible anymore. So it was something linked with the cysteine. And at the end, so what we could demonstrate also in collaboration with Change in Naples, that these compounds do alter the stability of the protein. Indeed, the limited proteolysis showed increase in the band after cleavage with the protease. And also they could prove that in the presence of in the assay condition, these compounds were causing a cysteine oxidation. And that was demonstrated by LabCMAS. And this was linked with the specific chemistry of these compounds that in presence of the reducing agent and the protein do create basically some ROS, some oxidizing molecule that interfere with the protein. This is the reason why it was denatured. I think I can conclude just to tell you a last story that heavily involved Marta in the last 3-4 years. Is the story of these glycogen binding proteins, that class of protein we were not aware of, and this started through a collaboration with the group of Graziano Lolli at Cibio in Trento. So PTG is it's a very interesting protein that has a role in the glycogen metabolism, as PTG stands for protein targeting to glycogen. And this PTG binds the PP1, that is a phosphatase 1, and there's a role in the whole mechanism of the glycogen storage. This glycogen targets protein or subunits are there are a number of homologs in the human. Many of them are concentrated in muscles and livers, but this PTG we have been focusing also is more ubiquitous, but we knew it is linked with a specific disease that is a Lafora disease, that is a dramatic epileptic origin pathology that it's linked with a genetic disorder, and that leads to formation of these Lafora bodies within the central nervous system, and affects very young people in the age of the teens, and that they have, at the moment, they have really no chance of escape, and there is no say any possibility to survive from this disease. It's a rare disease, but there is no cure at the moment. So we started from the beginning with the classical approach, that was that to study the protein. The protein has this CBM, that is a carbohydrate binding domain. And Marta has been producing this protein and obtained the crystal structure of it, observing in presence of cyclodextrin, that is, say, mimicking a sugar. She observed there were 2 different binding sites. That was a bit unexpected, and so we went through demonstrating that this binding site indeed in a system like in ITC could not be supporting the double site binding model, but while it's more probably an artifact of the crystallization, while we could measure using the wave system of the GCI Creoptics, we could measure the binding potency that was indeed in a range of a rational, a logic binding for a kind of, say, supporting protein. The story didn't finish here. Marta managed to obtain the protein in complex with the phosphatase, and in this case she generated a longer construct that was essential because the PP1 binds in this N-terminal region, and the crystal structure could be obtained. And with this, it was clear that while the carboxyl binding domain it's only located on a side, the rest of the protein, the N-terminus, is located on the other side, and the all proteins embrace the PP1. And this is kind of a key understanding of a specific binding point that is linked with a short peptide that is say, signature of the binding for the PPY. What was of interest is that through WAVE, we could also mimic the binding and measure the level of binding and understand also there is the peptide, using the peptide that can bind this specific region. We observed that has quite a micromolar binding affinity. To conclude, I mean, the whole story, this was, we completed the activity also using SAXS in collaboration also with Gabriele Giacchini in Padua. And this was a bit unexpected because at the end, SAXS is it's not easy to understand. The green part is where the SAXS is telling you that the CBM domain is located, while the crystal structure was different. And so what we observed at the end, the model is that indeed you have the PP1, the PLP, the PTG embrace the PP1 and has a module that moves around, probably doing his work in getting the glycerol and making the assembly of all together. Of course, this work is relevant because we intend to pursue, and there is a work done by Graziano in Trento, is to design inhibitors that can block this interaction and say, because it's reported that inhibiting the activity of CBM in this disease and as a proficient result. I conclude, I just mentioned a last class of protein, that is the ubiquitinases, that are historically been of interest in the lab. At the moment, we have been refocusing on USP1. That is quite an interesting ubiquitinase that is linked with cancer. We are progressing in preparing the protein with a sound of a number of escamotage. And so we are, when that protein would be interesting to be studied by classical screening of compounds, crystallography, and ideally also cryo-EM, because it has the size to be studied by cryo-EM, and there is already a cryo-EM structure from another laboratory who published 2 years ago this structure. Yes, I conclude. This was just to mention the future upgrade of the lab thanks to the project. So thinking to the possible collaboration, so basically we are experts in doing protein, so we can help the other in obtaining the protein that is of interest, and our focus, of course, is continuous on small molecule binding and development of protein target based drug design. And this is just to thank everyone. This is the present group that we have with some past members that left after the, either after the COVID and in some other, and concluding their activities. And there are a number of collaborators I have cited that have been involved in the project that we have been mentioning. So this is, I hope I gave you an overview.